Hi, good morning. I'm Holden Orius.
I'm Albert Gonçalves,
I'm Noah Schechter.
and we will explore about using fluorescent false neurotransmitters to map neural connections.
So we look to determine the synaptic level neural transmissions of serotonin in the brain, especially in the occipital lobe. And to do that, we will take live brain tissue cultures, and we will use a fluorescent false neurotransmitter, which is a fluorescent dye that mimics the structure of a neurotransmitter. So it can be imaged under under confocal or STED microscopy, which was discussed earlier. We will also excite a neuron to watch how the pathway works in that sample of brain using optogenetics, which is a field of exciting neurons using light.
So, after looking at our research papers and other sources that we found online, we decided to pose ourselves this specific research question, which is, what specific neural path does serotonin take in the occipital lobe of mice at the synaptic level? And the knowledge potentially gained from answering this question would contribute to brain mapping in mice. And since the brains of mice are basically scaled, a watered-down version of human brains, the knowledge gained from this could potentially contribute to the central goal of the BRAIN initiative.
And this is a long-term social context, but understanding the exact synaptic levels, synaptic pathways, through which serotonin travels in the occipital lobe of mice could eventually contribute to a larger understanding and mapping of the brain of both humans and mice, which at some point in the future, not only accomplishing one of the main goals of the BRAIN initiative, it can also lead to advancements in medicine and technology, which can save lives and money.
So to complement our work, we looked at some studies and we selected the most important information. Scientists are able to prepare live brain slices from adult mice for observation in a microscope. The fluorescent false neurotransmitters, the FFN, are currently being used to image and study synapses, and the transfer of dopamine has been studied using FFN in the past.
So after research, we decided to propose this for our project, which would be A, to introduce a fluorescent dye which would simulate the structure and function of serotonin, so it would be able to be passed along through synapses when neurons fire. Secondly, we would integrate the FFN into a living brain culture of adult mice, along with an optogenetic actuator. Now, you might be thinking, what the heck is an optogenetic actuator? And what it is, when you stimulate neurons using optogenetics, which is basically a field of science that uses light to get neurons activated, you need to introduce a specific gene that knows when the light's targeting it, so it'll activate the neuron. So we would optogenetically stimulate the neuron while it's under STED microscopy, and using this, we would observe the pathways of neurons as the FFN travels across each synapse. The advantage of using STED microscopy for this is that combined with double lasers plus a pinhole to block out excess light, we'd be able to get very specific, high-resolution 2D cross-sections of the brain sample all the way through, so we'd have a better map to analyze in the end. And in the end, we'd be able to map the pathways the false serotonin takes as different neurons are stimulated.
To reach the final objective, we first need to surpass some obstacles. Optogenetically stimulating a neuron under STED confocal microscopy is a new technology, so the knowledge about it, it's not quite enough to solve some issues that could appear while observing the sample. The STED laser and the confocal laser must stimulate unwanted neurons, what would not help in mapping the brain. And it's difficult to prepare slides for STED because they need to be really, really thin, and thin brain slides have a few neurons, and that could not be enough to completely map. Also, the STED laser and the confocal laser could have enough power to kill some of the cells.
So, in summary, the goal of our research would be to study the neural pathways of mice in the occipital lobe all the way down to the synaptic level, which hasn't been covered in too much detail before. And the project would use a fluorescent false neurotransmitter and STED confocal microscopy to track the firing of neurons releasing this neurotransmitter, serotonin. After getting the results, we'd be able to possibly conduct future analysis in other parts of the brains of mice to further create a comprehensive map of the mouse brain, and the results from this we'd be able to extend to human brains because they're very similar in structure and function.
So we all contributed to a variety of components of the PowerPoint, and we credit various scientific literature and other informational text.
Thank you. Any questions?
All right, questions? Yes.
Just like traffic here in LA, are there like possible bottlenecks in the neurons?
That's
Is that what you guys try to prevent, or?
we're not actually trying to prevent any certain disorder. What we're trying to do is map on the synaptic level how the serotonin is transferred between one neuron and the next, and how the neurons signal to each other, because that has never been done before for serotonin on the single neuron level. So we're not trying to necessarily treat a disorder that could come further down the road.
Is there a disorder that bottlenecks like the neurons? Is there like a bottleneck that causes a disorder that does that, or would that be in your field?
That's beyond the scope of our project in particular, so I don't know.
There's a question over here.
And over there.
Question over here? Yes.
Why serotonin? Where does it connect to the brain?
Okay, so when you're mapping neurons at the synaptic level, there have been many studies done targeting, say, dopamine and other neurotransmitters as such. But in the occipital lobe, one of the neurotransmitters that's more in action than dopamine is serotonin, and so we decided to target that instead.
Instructors, TA, questions over here?
I want to read your paper.
Yes.
You mentioned that there's a live brain slice.
Okay.
What
so,
is that? I've never heard that before.
So this is one of the things we actually managed to discover in the scientific papers we had. There's special ways to prepare slices of mouse brain. It's not say completely like alive and functioning inside the organism, but it's able to be preserved in culture long enough for the neurons to still have their function.
Thank you guys very much.